name is Ashley Voltz, and I'm with Antibody Engineering, and I'm speaking with David Younger today, CEO and co-founder of A Alpha Bio. Thank you so much for speaking with me today.
Thank you for the opportunity and very excited for the upcoming conference.
Yes, me as well. Let's get right into it. What are you seeing with the current state of protein engineering when it comes to AI tools? And what are some of the current limitations and bottlenecks you're still seeing?
Yeah, so with AI tools, we're seeing a very rapid improvement and acceleration in the prediction of protein structure, which has really launched the field forward and has been incredibly exciting. We've also seen a lot of fantastic advancements in approaches like de novo design for engineering novel protein sequences from scratch. Uh, but I think there also poses a limitation where we're not yet at the point where we can computationally design a protein in silico on a computer and expect that that individual design is going to work, right? We're still, even for kind of simpler problems against, binders to simple targets, sitting at, single digit success rates, single digit percent success rates. And for more challenging targets, those success rates go way down. So we're still very much at a point where experimentation is central to computational protein design.
Interesting. Okay. And why is large-scale experimental data so vital to these AI workflows?
Yeah, so large scale experimental data is both very important at the front end. So having training data to be able to kind of ground a model in kind of real world data. Ideally, it's data that is all coming from a common platform under common conditions so that it doesn't have kind of biases and other challenges with trying to kind of scoop together data that's generated in lots of different ways. Um, but large scale data is also very important at the back end, right? So once a model has been trained and it's made predictions to benchmark models and to validate designs that are coming out of models, we need to be able to characterize, not just dozens or even hundreds of designs, especially for those really challenging design problems. We want to be characterizing thousands, tens of thousands, hundreds of thousands of designs that are being proposed by AI models.
Okay, great. And what is A Alpha Bio's approach to integrating high throughput data and AI models for protein engineering?
Yeah, so at A Alpha, we have a synthetic biology platform that allows us to measure protein-protein affinities at a massive scale. So we can take libraries of antibodies, libraries of antigens, and measure all of the pairwise interactions between those libraries in order to kind of fully characterize those antibodies from a binding perspective. We then use that data to train binding models such that we can predict binding with novel antibody sequences, and then use those models that are now able to accurately predict binding to make predictions of sequences that are not just going to be optimized for binding, but also for developability properties and other kind of key attributes that are required for antibody therapeutics.
Right? And how have you used your platform technology specifically to engineer high quality antibodies?
Yeah, so we have used our platform in a number of different settings. So, for example, we have started with a parental antibody that is a kind of fairly weak binder, and in a single experimental iteration, engineered that antibody to be both potent to human as well as animal homologs, but also dramatically improve developability. So, essentially, in a single experimental step with AI as part of that workflow, have gone from a parental antibody to a very significantly optimized antibody that's a picomolar binder that also has significant mouse and cyno cross reactivity, and is very favorable from a developability perspective. So that's kind of a simple application of our workflow. But we also apply our platform in much more complicated cases. So, for example, working with Gilead in an HIV program to engineer a binder that's cross-reactive to about 1,000 diverse HIV strains to avoid escape to any of those strains and maximize cross-reactivity. And then much of the work that we're also doing internally focuses on engineering dual-specific antibodies. So single binding domains that are biparatopic and able to bind to two antigens simultaneously. And these are more complex antibody engineering challenges, but ones that are very well suited by our platforms.
Interesting. And what do you see as the future of protein design when it comes to accelerating drugs into the clinic?
Yeah, so I think that the use of computation can absolutely speed up discovery, right? So instead of having to spend, many months potentially doing animal immunizations or phage display campaigns or yeast surface display campaigns, the use of computation upfront can reduce that experimental time and therefore speed discovery. But I think that the gains that can be made in discovery are relatively small, right? Really, the time consuming part of getting drugs to the clinic is kind of all of the IND enabling studies up through clinical trials. And really what it comes down to is if we can engineer better antibodies, so antibodies that are incredibly potent, that have very, very specific binding for their target, where we really minimize any possible off targets or polyspecificities. If we can engineer antibodies that are enormously developable, then all of those downstream steps will be lower risk, and also faster. So really, I think it's less about kind of the act of substituting experimentation for computation to speed discovery, and much more about, can we engineer truly better antibodies, better drugs that are going to have both a higher success rate and faster timeline into patients.
Wow, very interesting step. Thank you so much for speaking with me today, David, and make sure to visit A Alpha Bio's booth on site at the antibody event in December.
Thank you, Ashley. I appreciate it.